On this one silicon chip, 64 tiny spots are each growing a different strand of DNA at the same time. The chip is choosing where to spark a little acid, spot by spot, and the only liquid sitting on top of it is water.
Here is the plain version of what you're looking at. DNA is a chain of 4 chemical letters. To build a strand to order, you add those letters one at a time in the exact sequence you want. The usual industrial way to do that runs on harsh organic solvents and specialized chemistry. This chip takes another route. It grows DNA with an enzyme, in water, closer to the way a living cell builds its own DNA. The team behind it works in Donhee Ham's lab at Harvard's engineering school, with Woo Bin Young among the lead researchers, and they published the result in Nature Electronics in June 2026. The enzyme they lean on is a natural one that stitches DNA letters onto a growing chain. But that enzyme has a stubborn habit. Left alone, it keeps adding the same letter over and over, so you lose control of the sequence. So the researchers use a trick. Each incoming letter carries a tiny chemical cap that blocks the enzyme from adding a second one. Add a letter, and the chain stops. To continue, you have to pull that cap off, and the cap only comes off in acid. That single fact is the hinge of the whole design. Control where the acid is, and you control where DNA is allowed to grow next. That is where the chip earns its place. Each of its pixels has 2 concentric metal rings and a spot in the middle where the DNA is anchored. Run a current through the inner ring, and it produces protons right there. It makes the water locally acidic. On its own, that acid would spread outward and leak into the neighbors. So the outer ring does the opposite job. It consumes protons as they drift out, mopping up the acid before it can travel. Inner ring makes the acid, outer ring fences it in. The result is a sharp little pocket of low pH sitting right over one strand of DNA and nowhere else. Now, scale that up. The chip is a grid of these pixels, and it can switch each one on or off independently. So the researchers write DNA in cycles. In each cycle, they pick which spots need the next letter, light up the acid at exactly those spots, strip the caps there, and let the enzyme add a base. Change which spots are acidic from one cycle to the next, and different locations receive different letters. Run enough cycles, and every spot spells out its own sequence in parallel. When they ran it, the chip built 64 different DNA strands, side by side, each about 38 or 39 letters long. That number matters because milder enzyme-based methods had struggled to make more than about a dozen distinct sequences at once. And when they read the strands back, the accuracy held up. In one full run, all 64 came out the right length, and 61 of the 64 matched the intended sequence exactly. Then they did something to make the point concrete. They took a short passage of text, about 169 bytes of it, and stored it in the DNA. Each pair of bits became one of the 4 DNA letters, so the message got split into pieces and written across the 64 strands. Sequence the chip's output, translate the letters back into bits, and the original text reappears. It is a small demonstration, not a hard drive, but it shows the chip writing real, arbitrary information, not just test patterns. There is a nice backstory to the hardware itself. The electronic backbone of this chip was not designed for DNA at all. It came out of the same lab's work on brain research, a silicon chip built to listen in on the electrical chatter of living neurons. The researchers took that dense grid of electrodes and retooled it, adding the ring structures and the acid control chemistry until the neural recording chip became a DNA writing one. But the most honest part of this story is where it stops working. When the team tried to pack the writing sites closer together and denser, the electronics kept doing their job. The chip still confined the acid to the right spots. What broke was the chemistry. The reaction that strips those caps seems to throw off other reactive bits that wander away from their spot, And when the spots are crowded, that wandering blurs which strand gets edited. So the limit here is not the silicon. The silicon is ready. It is the chemistry of taking the cap off cleanly that has to catch up. Which is a strange and interesting place to end up. The usual complaint about new technology is that the hardware cannot keep up with the idea. Here, it is the reverse. The chip can already switch thousands of tiny chemical reactions on and off, in water, in parallel. If chemists can find a cleaner way to pull those caps off, one that does not leak, then the same kind of chip could write DNA far denser and cheaper than it can today, for making genes, for testing biology, and maybe someday for storing data in molecules. The electronics are waiting. The next move belongs to the chemistry.